Okay, I hope you're not tired yet for the exciting part, which is using machine learning on pharmacogenomics data. So I was working in Benjamin Hepkine's lab as a postdoc until recently, that I started a new job as a machine learning scientist team lead at Cyclica. So since Cyclica is funding my attending to this conference, I'd like to talk about 5 minutes probably about Cyclica to introduce it, and then I will jump into our talk about machine learning in pharmacogenomic datasets. Cyclica is a Toronto-based company, in this company we are using artificial intelligence to do drug discovery and to reshape the process of the drug discovery to do it more computational. And we are working on two machine learning engines. One of them is Matchmaker, which we are using that to predict the binding of the drugs to protein, basically doing drug target interaction prediction. And the other one is Poem, which is a supervised machine learning approach. We are using it for predicting the pharmacokinetic properties of the drug. With having these two engines in hand, we are offering two products, Ligand Express and Ligand Design. Ligand Express is used for off-target screening, and Ligand Design is used for drug discovery, computational drug discovery, which speed up the drug discovery process a lot with very nice drugs in hand. We have some validation for this, and it works very well. So this is it was an introduction for Cyclica. Now I'm continuing with the tutorial. So in this section, I'm talking about evaluating the reproducibility, to answer your question about the reproducibility and the consistency between these data sets, and then about the meta-analysis and doing machine learning on these data sets that has been introduced by Peter to you. So as Peter and Arvin explained also, with these pharmacogenomic data sets, we have two types of data in hand. One is pharmacological part, which is drug response data, and then the other part is the molecular profile. And with machine learning part, we would like to be able to predict the pharmacological profiles from the genomic profile, any kind of genomic data, or any kind of profiling of the molecular data that we have from those cell lines, and then use them as the model to be able to predict probably patient's response. So with PharmacoGx, we have a lot of curated and annotated data sets in the same and standard way in hand. CCLE and CTRPV2 from Broad Institute, GCSI from Genentech, GDSC 1000 and other GDSC datasets from Sanger Institute from Finland, GRAY, which is only a breast cancer dataset. We have all of them in the same way annotated and curated, so we can do a lot of meta-analysis with these datasets now. So these are all, you can have access with PharmacoDB as Peter presented, and also with PharmacoGx, which is an R package that we are using to do our analysis with these datasets. So after we built this PharmacoGx package and annotated these datasets in the same way, we had this data in hand to be able to do some machine learning. So the first thing we did was to build a model. That's the first question come to your mind. If we can validate the model that we build with one of these datasets in the other, that would be the first validation, so that we can move forward with validating this in vivo, in PDXs, and patients. So we built the models using one of these datasets. We did with cross-validation in GDSC, and then we applied the model to do the test in CCLE, because they have about 15 common drugs, and they have a lot of cell lines in common. But with this kind of approach, we weren't able, the ratio of validation wasn't that good, and we weren't able to validate our models very well. So we were thinking, what is the reason why we are not able to have this validation? So we started to look more closely into the data. So I'm here showing two those response curves from PharmacoGx And you can see two cases, like for AZ is 6244, and applying this on a colo cell line, it's totally resistant in both data sets. One is shown in blue, one is shown in red. And then applying a plexicon on another data set on another cell line, in these two data sets show that they are very nicely, show very sensitive profile in both and very nicely correlated. But this is not always the story. Sometimes we have cases like this, like in one of the data set, the cell line is very sensitive, show very sensitive to the drug, and the other data set shows very resistant. However, it's not always the case. There might be some sort of inconsistency in the cases that we observe. So this is something that we, from these, like from when we're talking about consistency between these data sets, that's something that what we expect. Like we expect to see some cases of resistancy, which are the points that I'm showing here. So through this slide, I always use AAC, which is area above drug dose response curve, like the area above this. And if it's 0, it shows resistance, and if it's close to 1, it shows completely sensitive. So those red points, they are resistant. Those blue points, we expect some sensitivity, and then we expect to have some inconsistent cases like the green and the orange dots that I'm showing here. This is something that what we expect, but this is something that we observe. So you can see that with, I show for paclitaxel 17AG, and AZD6244 and PD03 to 25901. So we have a lot of green and orange points. So it's very hard, if that's what we observe, it's very hard to build a model to be, to show a very good validation in our test set. So how we can handle this? So we started to we have a very long history on how we try to handle this. So we started to use different methods to assess this consistency, like Spearman, all the common methods that may come to your mind, adaptive Matthew correlation coefficient, many methods we used. And then that's what we land on. So we learning from the survival data, and we had experience with, and we had experience with using concordance index. So we found that it's one of the metrics that work very well for assessing consistency here, and we can adapt it to tackle the noise in this data, and I will explain it in a few later slides too. So first, if you are not that familiar with concordance index, I put some introductory slide here. So if we have these two vectors and we want to assess the consistency, the way concordance index assesses the consistency between these two is by considering all the pairs in these vectors, and then we consider for the other vector if the order of the pair changed or not. So like if you look at Y, all those red arrows show that the order changed, like it was yellow before red in X, but now red is before yellow. So if you want to, and then the black arrows show that the order stayed the same, even though the value might have changed a bit. So we don't care about value, we only care about the order. So the way we assess the concordance is by counting the number of concordant pair and then divided by all the number of all the pairs, and then the consistency between the concordance between these two, or consistency with these two, is 0.67. So with this metric, we can have the different values. Like if it's 0, it shows that they are correlated, but in inverse way. If it's around 1, they are completely correlated, and if it's around 0.5, they are their order is completely random. There is no correlation at all. So now let's look at how we use this in assessing the concordance in pharmacogenomic data sets. Here I'm showing all the biological replicates that we have across all those pharmacogenomic data sets that I showed you that we curated in PharmacoGx. we have around 19,000 biological replicate, and by biological replicate, what I mean is that in the same center, they did the high throughput screening with this, on the same cell line with the same drug twice. So here is the distribution of these biological replicate that you see. We expect that they were there because they are biological replicate, they are from the same center, so you expect that they are very correlated. But you can see that they are still a bit spreaded around the, like you have, there is a very good correlation, but there are still cases that they are not very correlated. So with, if we use the concordance index itself, the concordance index that you see is about 0.78. If it's like it's our highest concordance that we can reach because it's from the biological replicates. But that's not what we expect. We expect something very correlated to be our highest. So we introduce RCI, robust concordance index, that can reach to 0.92 for this biological replicate. So the way we developed RCI is by looking the distribution of the difference between these AAC values in biological replicate. As you can see, I plotted it on the right side. And you can see that it's very well centered around 0 and with a standard deviation between -0.2 to 0.2. So you can see that the noise in biological replicate is something between, if I consider the 95% of the cases, they are all stay between -0.2 to 0.2. So if we consider that as a noise in these kind of assays, and so we can say that for if the two AAC values are different, that their difference is less than 0.2, we cannot say if they're the same or not. So we remove these cases as invalid and only consider a comparison as a valid comparison if their difference is greater than 0.2, and then that's how we are dealing with this situation. And then we can reach to 0.92, which is something that you expect from high profile institutes, that they put nice data together, which is something that is happening probably. So here I'm showing that how RCI improved the consistency that we can assess by looking only at the observed drug response across different data sets. I look at the 4 different data sets, CCLE and CTRPV2, as I said, from Broad Institute, GCSI from Genentech, and GDSC 1000, and I assess their concordance using CI as shown in gray box and RCI as shown in the red box for any 2 of these 4. So you expect that you would see something 6 points in each of these boxes. And then you can see I sort of ordered these drugs that they're very targeted therapies to more chemotherapies, and you can see that how RCI improved for targeted therapy. The concordance is for most of the cases above 0.8, and it's very nice. But for cases like the 5FU, and if I have more here, for chemotherapy, it doesn't improve much because it's still very spreaded and the noise is so much more that we cannot concur with this kind of universal approach that we, universal delta that we considered as around 0.2. So, but still it's very nicer results that we can reach with this new metric. So here I'm showing how we are doing biomarker discovery. You can use this concordance index for assessing the association of biomarker one biomarker to drug response, and then we use a random model to do the meta-analysis. And so you can see I show for for this biomarker in CCLE, GCSI, CTRPV2, and GDSC, the concordance index, which is RCI, and the RCI and CI both, and then the p-value, and then here you can see the effect size, which is the actual value for RCI, and that the error bar shows the standard error that we have for these. So yeah. So here, if it's toward 1, it shows that our biomarker is predictive of sensitivity. If it's toward -1, it's predictive of resistance. And then, as I told, the point is the effect size, and then the confidence interval around it, and then the p-value shows the significance, and then we can do the meta estimate. So you can see here, for example, for EGFR, which you expect copy number variation or the amplification of EGFR to be predictive of response to Erlotinib. I did the analysis for both copy number variation and for the expression. And you can see that how with RCI, like our assessment improved a lot, and especially that you can see that how we can capture the signal with mRNA expression much better than copy number variation. And for the mRNA expression, I use RNA-seq data, which also is more consistent across data sets and showed the signal much better. So you can see that how, like in some cases, the signal was completely diluted. Like for CTRPV2, I guess it was around 0.5, but with RCI, it improved a lot. And when I'm talking about RCI, we only apply it on the drug part. For the expression part or for the biomarker part, we are not applying any delta. We consider all the pairs that are valid. So now it was about univariate biomarker, that how we can use a method like RCI, or a method that I'm showing you in the tutorial as well, that we have already implemented in PharmacoGx. But you might want to also do multivariate modeling, and you can use different methods to do that. For example, in CCLE and GDSC datasets, when they published their paper in Nature in 2012, both of these papers, they use elastic net for multivariate models. As input, they use, in GDSC, for example, they use the mutation profiles of over 64 genes, the gene fusion that they had. They use the gene expression of about 10,000 genes, and also the tissue type of all the cell lines as input, and then they try to predict the IC50. And then they did a 10-fold cross validation, 100 times shuffling for that. So I will show some sort of this implementation, how you can do some analysis like this using PharmacoGx in the tutorial as well. Also in CCLE, they did something very similar, and they tried to predict IC50, Amax, and AUC, all these three. And then also they did some naive-based classifier using the discretized values. So you can do many different machine learning methods. I will show you a few cases that I implemented, and you can think of other methods that you can implement. Also, I'd like to talk about more complicated methods that people might have developed, and you might have idea about developing them. There was a NCI-DREAM challenge in 2014. In the challenge, there was a training set of 35 breast cancer cell lines. For that, the team provided mutations, CNV, DNA methylation, gene and protein expression for 28 drugs to all the participants. And the participants were supposed to provide the response to predict the response for 18 other breast cancer cell lines. For those breast cancer cell lines, they also had all the profiles, but they didn't have access to the drug response. But they were assessed in by NCI-DREAM, but they didn't have access to. So the best the top performing method was by a team from Helsinki University, and their method was to, they provided a method, which in that method they used 4 machine learning concepts. They did a multi-view learning, meaning that they used different view of the data plus all the profiling that has been provided to them. They try to look at the pathway data, different views of the data. I think they looked at 22 views of the data, and also allowing the continuous value for each of these views. They also did the discretization of the data. In the A, you can see that these views have been used as input for the model, and then they use different kernels. They use a kernelized regression instead of the standard regression methods to be able to capture the, instead of using because of we have a lot of feature in these data that you can consider, you can imagine we have 40,000 genes that we are assessing the RNA-seq data. So they wanted to make the parameter space smaller with the kernelized approach. They use these kernels and then they integrated all the kernels, and then also they try to do a multitask learning by having these models to talk together when they are predicting the response for different drugs. And then they use also Bayesian inference because the data was so noisy. They wanted to have some probability to assess the models. So this is a very complicated model they worked on, and it was the winner of the NCI-DREAM. But you can think of simpler model to do also machine learning in these data sets. So one of the, also I want to talk about some applications that we worked on with PharmacoGx. One of these applications was to, with having this RNA-seq data in hand, people are usually using gene expression or gene mutation or copy number variation as predictor of drug response. We wanted to look at isoforms and see how it's capable of to do better prediction of these data. And we published this in Nature.com 2 years back. So what we did was to use both CCLE and the GDSC drug response data. At the time, we didn't have RNA-seq data from GDSC, but we wanted to do some meta-analysis to also tackle this noise data. So we were using both these data for the training set. And then we used the CCLE isoform data also as the features. We did multiple validation using gray data sets and GCSI. For GCSI, we were able to do a pan-cancer validation, and with gray, we were able to do some breast-specific cancer cell lines validation. And then our standard, our final validation was using UHN. That was a data set that we were putting together in U of T ourselves. So here I show some of the results that we had. So these are, so for Erlotinib, these are the best predictor that we had. All of them are isoformic. So if we had a gene-specific biomarker, I would have marked it with as red, but for Erlotinib, we didn't have any gene-specific. So we were assessing both genes and isoform, and if gene was overperforming isoform, we would have reported that, and if isoform was overperforming, we were reporting that. And you can see that you expect that with, if the isoform is predicting the response for being sensitive, you expect that you see a reddish part on the right, and then for the resistant cell lines, because the cell lines are ordered by their sensitivity, you expect to see some bluish on the left, and that's what you see. Even though the data is noisy and the signal might not be that great, but still there are some signals there. And so I in the tutorial, I'm talking about a way to do some feature selection, because as I told you, the data input that we have for molecular data profile is very high dimensional. So we want to do some feature selection to not have overfitting when we are doing the machine learning part. What I use for feature selection is the MRMR, which means we choose features that have minimum redundancy, like they are less correlated together, and they have maximum relevance to the output. So there is a package in CRAN for MRMR, has been developed by my supervisor Benjamin and his group at the time, and you can use it. And so it's in the tutorial available. I will show you. And so here I will show you I did some ridge regression, and then I did some cross-validation shuffling, and we do the other methods for machine learning, for multivariate modeling as well. So for ridge regression, here I used the CTRPV2, like I only wanted to do CNS cell lines. For cross-validation, if we do 100 times shuffling, the error bar shows the shuffling, and then I use RCI to assess the models. You can see that, like, at least for, I did build the model for 90 drugs here, and you can see at least for half of them, we were able to build some good models that are applicable if we want to proceed to test them in the other models or in the patient or something. So here is some of the applications of PharmacoGx, and with that, I'd like to move to the tutorial. Any question? The question was that when we are using RNA-seq data, if we use the continuous values or we discretize the RNA-seq data, but we use the continuous values here. So here the first chunk shows the packages that we need for this tutorial. So they are almost the same as the ones that we need for PharmacoGx introduction that Peter present. So we need PharmacoGx package and BioBase package, which is one of the requirements for PharmacoGx to work with molecular profile. And here also you need to install 2 packages from BHK Lab, because one of them is WCI, which implement that RCI assessment that I talked about. We haven't published it yet, so it's in, but it's publicly available. You can install GitHub and have access to it. Also, I developed a package to do this machine learning part for the workshop, which is PharmacoGx ML. This is also publicly available from our BHK Lab GitHub. You can have access to it and install it. In this package, I do some machine learning with random forest, SVM, and ElasticNet. For these 3, you need GLMNet, Caret for SVM, and random forest for the random forest machine method. So these packages also you need for that. And then this other package is for a bonus part that I will talk about is at the end if you have time for it, or if we don't, you can do it later. So this part is, I download CCLE and GDSC as Peter explained. I don't think I need to do it. Probably I have access to it. I don't think you need to do that neither. So for the next part is where I do assessment of the consistency between these 2 data sets. So first we need to get all the common drugs between CCLE and GDSC. So I do an intersect p-set and then intersect on cell lines and drugs between CCLE and GDSC, and then we need to do summarize sensitivity profiles and, to get the AAC values, because like in CCLE and GDSC dataset, you might have different profile available for pharmacological profile available for these pieces. We have AUC, we have IC50 recomputed and published. We also have the published data available, but here I use the recomputed part ones because I want to be more consistent in the way we fit the curve, because they use different ways to fit the curve. And then then I do use WCI package that I explained to do compute the concordance index. So you need to pass two vector to it for that you want to compute concordance between them. And then if I put delta pred equal to 0, it does the conventional concordance index. And then if I put 0.2, then it does this RCI that I just explained to you. And then at the end, we plot the bar plot for this. So if I run this chunk, you can see that it gives you this plot that it shows in red the RCI and in blue the conventional CI, and you can see how it improved for most of the drug, the concordance between two data sets for each drug. So for the next section, either you can use RCI as I explained here, or it's not yet part of PharmacoGx that we use RCI for assessing the concordance, for assessing finding this association of biomarker to the drug. But we have another method there, which is a the regression model. In this regression model, we use the tissue type and gene expression value, or any kind of profile that you have mutation or gene expression value. And then B is also if you have any any batch effect. If any batch effect is available, you can also add this to the model as well. And then the bias. So here I show how to use this to assess this univariate association. So you need, so I wanted to do it for EGFR and Erlotinib for expression, and then BRAF mutation with PD 0325901. I know that these are predictive of the response, so I want to see if I can see this prediction in the data sets as well. So I use RNA data because I wanted to see it in both CCLE and GDSC, and we only have microarray RNA expression data in both of them. You need to get the feature because the features, as Peter explained, in RNA expression, they're ensemble IDs, and I wanted to use symbol ID to get the ensemble gene IDs. And then we use this drug sensitivity signature, which you need to pass the PSET name, the molecular data type that you want to do assessment with, like I pass here RNA, the name of the drugs, it could be multiple names, the name of the features you want, and then sensitivity measurement you want. And then in case there are replicates in drug sensitivity or replicates in the molecular data, which would be the case sometimes, for RNA, I use median to do the summarization, and for the drug also, because it's continuous value, I use median. For the mutation data, we use for the summarization for molecular data, you can use and or. For each mutation, you might have different mutation hotspot for that gene, so you want to consider either one of them is enough, or you want all the. And then I just summarized this signature in one matrix and that I want to show. And at the end, I plot them like box plot for the mutation and just a scatter plot for the expression. So let's run this chunk and see the results. Maybe I should run the next chunk also earlier. Okay, let's. should not be that slow, but I think. Okay, while it's running, I explained the next part, which I should have started to run before because it would take a while. So for the next part, which is multivariate modeling, as I said, I'm using the PharmacoGx ML package. For that, I'm using as a training set the GDSC data, and then I want to do the test on CCLE data. So we have, there is a problem here. Probably you would have that, so you better put this PharmacoGx in front of, because this function, we have it also in Jiva package that Arvind presented, so it gets confused which function we are calling, so you need to put the name of the package always. It's better. So I run it again. So I use for training, I use summarized molecular profile. Also, Peter presented to get the RNA data for all the features, all the genes that we have in CCLE. I use summarized sensitivity profile to get the AAC value to a lapatinib. I want to build a model for lapatinib in CCLE and then test them in GDSC. So for validation, I use sorry, the other way. For validation, I use CCLE, also use the same functions that I explained to get the molecular and drug sensitivity values. And then I have two methods in PharmacoGx ML. You can look into them. So the optimization, do the training. So you can pass the name of the models you want. I put the methods in, like, I want Ridge, Lasso, Random Forest, and SVM. So for Ridge and Lasso, I'm using GLNet, and then for Random Forest and SVM, also, we have some results, I guess now. Let's look at them first, and then I will proceed. So here is you can see that the effect size from the linear regression and the p-value, and you can see in both cases for both CCLE and GDSC, it's significant. So here is one of the ways to assess these associations, and then you should have some scatter plots and box plots. So the first ones show for CCLE and Erlotinib, the scatter plot, and then the box plot, you can see the significant signal, especially in the box plot, it's much more obvious. I think it's because it's rectangle, it's not that obvious that there is a signal. Okay, so I run this chunk, so it gets run when I'm going through the function. So you can determine how many fold you want for cross-validation, and also how many shuffling you want to get those error bars that I showed you in the presentation. But here I only do one sampling and 5 fold because it gets too long to do more. But in reality, you should do more and have the average of all the shuffling to not be biased toward the order of the samples. And also for feature selection, I use MRMR, and then I want to just choose 10 features for now. It's not a correct way, but I just for the aim of the tutorial, 10 features, because the other way it gets really long to run all these methods. And then, we have an error. You don't have MRMR? No, you didn't load any. Oh, yeah, you're right. Yeah. So first run the first chunk and then run this chunk. Okay, and then you want to know for the assessment, you can use either those common correlation method like Pearson-Spearman or RCI, as I determined here. And also for the validation, I report the model, put them in res, and then you can pass the model that you want to do validation for. It would contain all the, like if I use the model for lapatinib, it would contain all the models for all the methods that I passed it through their names. And then the validation set would be the molecular profile of the GDSC, and the validation label would be the actual label that we have in, for CCLE, and then actual label that we have in CCLE. So we want to do assessment for the actual label versus prediction using RCI. So you can change these parameters. And then it gives you two plots, one for the training phase and then one for the test phase. And you can see how it works in the cross validation in the training and how it gets assessed, and then do like some choose which model is the best for your case. So it get finished soon, I guess. Yeah, I can do that. But I think it's good. Oh yeah, it might not. It's still doing the lasso, so it might take long. So maybe we can see the results here. It opens it here. Oh, yeah. Yeah, it's working in comparison. Okay. So, yeah, you can see that like the RCI for reach, it was 0.82 in the cross validation, but in the actual validation, it's lower, it's 0.65, but actually it's one of our best. Like you can see the same results, almost the same for lasso. For random forest, it was a bit worse in the training, but it's less overfitting, I would say, because it's more like the RCI is more closer between validation and training. And also almost the same for SVM. But it's only a very small case. You cannot judge if it's really which of these modeling is the methods is the best for this drug with this setting that we had, because it was very low number of features that we used, and number of shuffling, it was 1. It's not enough. But I can tell for lapatinib bridge is kind of the best to capture. Also, they showed that in the NCI-DREAM that for different drugs, you would have different power. Like sometimes you can build a very good model for the drug, but sometimes you cannot. And it doesn't have much to do with the range of the drug response also, unfortunately. So it's a mystery. Kind of sometimes. But usually if you have a nice range of the drug response, it's the best to build a model. That's not always the case. So for it's not here in the built one, but we have a bonus section also. In PharmacoGx, we also provide functionality to work with perturbation data. So all these that we presented here was to work with pharmacogenomics sensitivity datasets, but we have there are perturbation datasets like CMAP that we also cover in PharmacoGx, and you can look how the expression of the data before and after treatment change, and build a signature to predict drug response using that to see if the drug could be effective on the cell line or not. So here in the bonus part, I use the signature for HDAC inhibitors that has been published already, and wanted to show that if we can capture that signature in the CMAP, and we can, if among all the thousands drugs that have been was in the CMAP, if those HDAC inhibitors with this signature comes up as top predictors. And if you go through it, you can learn how to use, so we have connectivity score to compute the connectivity score, to build the signatures. So you can use download perturbation signature to download CMAP, and then use connectivity score to build the to compute scores. And then if you, at the end, if you look at the HDAC ranks, you can see that these HDAC inhibitors, these 4 drugs, will show up in the top 5. like they are 1, 2, I guess another drug is rank 3 and then 4, 5. So yeah, it's okay, the last part is finished, I guess, but this part is also gets a long time to get finished, so you can do that later, I guess. Any question?
How many features were used in total?
Feature in which?
Like during the training process, how many features did you use?
For this tutorial, I only used 10 features, but usually you want to have a couple of hundreds at least features to capture the variance of the feature that might be predictive of. 10 feature is not enough for a whole genome-wide, if you want to look at, because genome-wide we have 50,000 genes, and then many of them might have different contribution to the drug response. So if you want to capture good variance of them, I think a couple of hundreds, like 100 at least.
And when you did feature selection, did you see any particular type of features that were more prevalent than the others? For example, if presence of some mutation or some RNA expression values were more important?
Yeah, it could be, definitely. Like sometimes mutation is the best predictor of the drug response. Sometimes it depends on the drug and it depends on the contribution. You can definitely do a combination of different feature type as well. But here for the tutorial, I only did like gene expression. I didn't even look at the combination of the different like genome, but it definitely is the case. And as I showed you also in NCI-DREAM, they showed that like they look at different multiple views, which come from mutation profile, amplification, expression, and also if you combine them in pathway and this, it could be like in some cases, one pathway could be the best that predicted drug response. Sometimes the mutation is the best predictor. It totally depends on the drug and on the situation.
Okay, thanks.
So also maybe to elaborate on that from the DREAM challenge, what they saw was that across the drugs that they were testing, on average RNA sequence, or it was, sorry, array. RNA expression was the single most informative data type, and followed by, I believe it was protein expression.
Yeah.
And that's sort of been recapitulated every time someone's done both these profilings. But that might be because we just don't have a good way to screen the whole proteome yet, while gene expression is giving us sort of this unbiased snapshot. So with gene expression, we can do data-driven feature selection. With protein expression, it's basically what we're able to measure in a high-throughput manner. And sometimes actually one feature is enough. So for 17AAG in particular, the NQ1 expression of the NQ1 gene has a concordance index, depending on which data set, but not a robust concordance, just a normal concordance index of around 0.8.
It's the best predictor among all the drugs, all the features.
Yeah.
It's kind of the best predictor of the drug response.
So really, and that sort of goes back, if anyone's ever studied machine learning here, the no free lunch theorem, and like you never know what's going to work in your particular case, which is why test different numbers of features, test different methods, and just that's why you need these big loops along these different hyperparameters.
Yep.
Sorry, another question?
Yeah, so everything gene expression is continuous value, that's why it's best predictor compared to mutation data. Okay, so it has been reported many times that gene expression is the best predictor. So do you think because it has continuous values as compared to mutation data where we get only binary values?
I don't think it's the only case. Like for also comparing, as I showed you, for EGFR amplification, which is expected to be a very good predictor of drug response to Erlotinib, still mRNA expression of EGFR is working better, like it's a better predictor. While for amplification, like for copy number variation, we also have the continuous values. But I think that probably the the assay that we use for expression data, they're much, the quality of the data is better, and also, I don't know, probably that's the case. And yeah, that's true that we have more features also with gene expression, but that's not the only case.
You mentioned that the best concordance index that you achieve is 0.8, which is relatively good compared to the baselines and the comparison partners, right? But how useful would that actually be in practice? I mean, how accurate, how reliable are such predictions in practice? Is a physician ever going to use such a method to support his or her decision-making in practice?
That's a very good question, and it's a very long line that we have, I guess, to translate these findings to clinics, and it's really hard to interpret these results in clinic, I guess. But it's at least since we are talking about the order here, and we are not talking about the actual values. It's more interpretable, I would say, in clinic than if we are talking about the actual values. But you have better answer for this?
I mean, I think it really depends on what, for that, let's say a particular cancer, what the baseline is currently at, and sort of at which, if the patient is out of options, in terms of especially some of these more, I guess I would say, novel clinical trial designs where they're enrolling across different tumor types, very late stage metastatic patients, and trying to do sort of data. Primarily right now they're focusing on clinical data mining, but doing matching to drugs like the IMPACT study based on biomarkers. And I mean, as a standard of care, we're very far away, but at some point, if there's no other option, I think it's still a useful task to work on.
Yeah, so I just wanted to more expand what Peter was saying. So we are from also part of the UHN University Health Network, which is the medical branch of University of Toronto. And we work a lot with the clinicians, physicians who are treating patients. So what we are talking is mostly about research or in the setting of the research. But when you talk about the clinic, it's completely different story. It's a completely different beast because you cannot give a patient a drug which is not FDA approved. So you have to go through all this cycle of the approval and all this. There are biomarkers, known biomarkers or well-established biomarkers of the drugs which are currently in use in clinic, where you have mutation or copy number variation, etc., are used as a biomarker for the selection of patients. So I think I showed some slides also. As Peter was mentioning, in our institute, through this research, we are more focused into basket clinical trial. So earlier when you do, or normally when you do clinical trial, you just select the population, let's say 100 cancer patients, and give your drug. But now field is more moving towards basket clinical trial. You have one drug that you developed for one particular mutation or one particular gene or pathway, then you select the patients which have those pathways abnormal. Okay, and you compare it with the standard of care. So these kind of biomarkers help us to select those patients. So you can select among your breast cancer patients, you can select the patients, let's say, which have EGFR pathway abnormality, because you are seeing in the cell line or in PDXs that your drug is more effective to the EGFR pathway or EGFR gene expression, just giving a random example. And then you can test or you do clinical trial only on those patients. So this is one of the ways that we are using it in translation. Another thing that we are doing is that we have in-house CBio portal, and this PharmacoDB and all these tools are also connected with this CBio portal, which helps the clinicians. So there is a tumor board which decides for a patient which medicine to give. So they use this information when they take their decision into account, whether they see some indication of a novel drug or a new drug, it should be FDA approved. But if they see that, okay, this particular drug is showing some indication in the lung cancer cell lines, though it's used in the breast cancer traditionally, or it's used in pancreatic cancer, but it's showing some good indication in the lung cancer also. So physicians might decide to give the lung cancer patient that particular drug. So more going to tissue agnostic. So this is another setting where these kind of method helping. Yes.
I have related questions concerning the discussion, so about this clinical translation of the pharmacogenomics data. So I wonder from your comparative analysis, whether you find a particular class of drugs which show relatively higher consistency across different studies, which might indicate of this translation in the clinic. So for example, if you look at some kinase inhibitors, normally you're expecting that what you see in the cell line sensitivity can be translated into the patient. So my question is that whether you find a particular subclass of compounds which show in general higher concordance index, as you have discovered, which can be present to the clinicians to see, okay, this is good, this drug might be translatable to the clinical practice.
So in general, from my knowledge or experience, usually chemotherapies are really bad in terms of biomarkers, and targeted therapies are good. You can find biomarkers for the targeted therapy because they're designed to target a particular protein or a particular gene or a particular mutation, so you can get a signal. Chemotherapy is more or less like throwing an atom bomb in a cell, and it just kills randomly something. But I think Zhaleh would be able to comment more into that in the cell lines. So what was the question is that when you do the consistency studies, do you see certain class of drugs more reproducible compared to others? And more into patient also.
Yeah, definitely. Like the target, as I also showed you in the validations that I showed, the targeted therapies are more reproducible, definitely, than the chemotherapies. And since the results, like these chemotherapies, they act weirdly when they treat cell lines with them, like they can show very variant results, and the actual value is not interpretable and it's not reproducible. So targeted therapies, they show more, like at least the resistant versus sensitive are more separatable, and so in that sense, they are more reproducible as well.
I just want to interject, but what might be a better or a different question that should also be asked is, I build a predictive model on my cell lines and I want to test it on patient data, but how conserved are the feature spaces that I'm predicting from? So it might not be a drug class, but there's questions like what Arvind was showing about even just conservation between PDX passages. There's some pathways that are unstable in these model systems and some pathways that are relatively stable. And sort of especially if it's, so two things I want to say. This is an under-investigated area, at least in my opinion. We don't have a definitive list of genes that if you find a biomarker in a cell line in this gene, it will or will not translate to let's say, an in vivo model. It's a complicated question. And then sort of the baseline, if it's not even stable in your own model, so if you sequence your model twice and the expression of those genes changes, that's a very bad sign. So or if you do passages of your model and the expression is very variable between passages, or the mutation, if your gene is often acquiring mutations as you passage, that's also not a good sign. So that sort of establishes the baseline of which genes you should not look at.
Okay, so before you ask your question, I guess I show you the results because we had time. So through this question, I ran also this bonus part for connectivity scores. And so for this, we use these genes. Like some of them, in the signature for the HDAC inhibitors, they're upregulated. We put 1 for them. If they are downregulated, we put -1 for them, and then we use them to compute the connectivity scores across 1,288 drugs. And you can see that these four drugs, they showed up as in the rank of 1, 2, 4, 5 as top 5. So if you run it, you can see the same results. Okay.
Right, so I just wanted to probably rethink the question that you asked. So because you guys have experience working with cell lines databases, so pretty much looking into past what has been characterized thus far, PDX, what it can in terms of patients, and maybe some clinical trials data. So do you guys have any maybe zooming out, but some sense of what are the features that are generally preserved? Or even that is too much of a generic question to be asked?
I guess, so if I understood correctly, your question is, what are the features that are preserved in patient to model system? Yeah. 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 So from our experience, our understanding till now is, or more or less we kind of assume, is as a truth, there is patient, then there is in vivo model like PDXs or organoids three-dimensional model, and then there is cell line. This is kind of hierarchy in terms of distance. That's what we believe. Certain papers have shown this also. For example, in lung cancer also, one of our collaborators have shown that the PDXs and patients are more close and the cell lines are a little bit far away. In terms of individual features, so there are certain growth-related pathways which are highly preserved across these models. So pathways which are involved in cell growth and all this. However, it's kind of it's still a very difficult question in the sense patients is vast. Every cancer patient is a little bit different, okay? And what we make the PDX or cell line represents a small subset of those patients. The success in PDXs, in organoids, or in cell lines is quite low. Depending upon the cancer type, in breast cancer, it's maximum people have reported up to 50% success rate. So out of 100 patients, you can make PDXs only in 50% of patients. Then again, the question is, there is a genetic drift passage to passage, whether in cell lines, in PDXs, or in organoids. So when you are creating these models, then you are getting the genetic drift. But in terms of mutation, it's quite preserved, or it's kind of used as a gold standard. If you're creating a PDX or organoid or cell line from a patient and you don't see the similar mutation profile, which are frequently mutated in these model systems, then your model system is basically useless. And mutation is the easiest thing to measure. In terms of gene expression, the problem in gene expression is very dynamic. It changes from time to time. I mean, if you are creating, even within the patient, with time, the gene expression might change, So it's difficult to say how much it's preserved or how much it's not preserved. Copy number variation, it's kind of in the middle, but for certain pathways, I think I haven't shown these slides, but I have a talk also. At least in the PDXs, we have a list of pathways and list of genes, which we have shown that it preserved from passage to passage, not patient to PDXs, but across different passages. And we have shown that there are certain pathways which are highly unstable. So we have this list. We are working in this direction in our lab and looking more, trying to establish more the pathways which are highly unstable and highly stable also. But in general, growth-related pathways are known to be stable. The question is, what about the immunotherapy? Immunotherapy. So cell lines, there is no hope in terms of immunotherapy. I mean, it's very difficult. People are creating immune mice or synergic mice. Basically, there are certain groups which are working to establish immune system also. In PDXs, there is another technique where you transplant the tumor as well as you try to transplant the immune system in the tumor, so, in the mice, basically. But it's highly complicated experiment because you have to do surgery in the mice and put the cells, immune cells, in the spinal cord of the mice, which takes basically quite complicated, and you have to have a good synergy between these two surgeries or two experiments in one mice, and it's very difficult to do high level or multiple, basically, or high number of these kind of experiments. But people are trying PDX model in that context. I'll say that there is really less hope using cell lines or organoids for the immunotherapy. People more love to work with the patient directly in case of immune therapy because, yeah, human immune system is quite complicated and there are a lot of different factors involved in the immune system also. But yeah, people are trying to develop cell-based systems when you have very specific antibody or very specific target that you want to hit. People are using, but it's more research purpose rather than finding biomarker. Yes, definitely. There should be some immune biomarkers, but what I'm trying to say is that people are using these model systems, cell lines, PDXs, etc., more for the research rather than doing large-scale correlation analysis or associating gene expression with the immune drug response, because that's quite difficult. Or these models are not useful. So basically, you can just look into one marker and in a cell line or one particular protein and then see binding of your immune-related drug or your antibody. You can do those kind of experiments, but not for the biomarker discovery, the model systems. People prefer to use more human systems or patient directly. So I would like to just give some concluding remarks. So as I said earlier, these were our objectives. We talked about different preclinical models. So I hope now you know about these preclinical models, the differences, their limitations, and their usefulness about that. We talked about how you can use these different data sets that are available and how we can use them for the pharmacogenomics, for the analysis. We perform biomarker discovery. Peter covered this part. Zhaleh in the last covered about machine learning and how to build these models and how to use them. Please, again, don't forget to do the feedback form. These are some of the references. It's available in the slides also. I would like to acknowledge the whole BHK lab and Microsoft Canada and Microsoft Switzerland. Thanks for providing the cloud access that you are using through the Azure platform. The work is funded by a lot of different organizations, OICR, Stand Up to Cancer. I missed the Vector Institute here also, and Princess Margaret Cancer Research Foundation, and so forth. And yeah. These are our email addresses if you have questions, and we are available here for today, tomorrow, for the conference. You can discuss with us. And just quickly, some people asked how to download it. So what you do is that just go to the RStudio here in the home and select ISMB and slides. Don't select the R, it's just libraries that you install, default location. And then, where is it? How do I move it? Yeah. If you click on here more, you can click on the export and you can give it whatever name you want. So it will download the whole zip file, which includes all these scripts and all the slides that we talked. And the feedback, again, the feedback link is available in the slide here. So you can directly click on the introduction slide, for example, and it will open the PDF. And I guess the last, sorry about this second slide, we should have removed it, but up. Okay, Peter, you have to. Yeah. How do I go to the end? Is there a command? So if you go to the end of this, here is the link, the URL for the feedback. Please provide the feedback. It helps us to improve this whole tutorial. And a tutorial doesn't make you expert in a field, but it gives you the tools and techniques and the basic information that you can use further. So in this whole tutorial, we have used example data sets. We have used really less number of feature machine learning and other purposes. But in most of the scripts, you can just change these numbers and run it for larger data sets. Most of the tools have by default parallel processing. So if you use the supercomputers, you can change the number of CPUs and then you can run it faster. So yeah, so you're welcome to discuss with us. I guess we are almost on time, so thank you very much. Yeah.
Also, we'll as soon as I get my laptop back, I'll click the button to make the repository for this public as well.
Oh,
and
yeah.
that's an easier place to download it. It'll be BHK Lab is our lab repository, and the name of the repository will be ISMB-2019.
Yeah.
Pretty easy
Yeah.
to remember. Yeah, and any things that small typos we had to fix will be fixed.
Sorry, I have made it public already, the GitHub also.
Oh, you have?
Can you show it?
Yep.
Yeah, I have made it public.
I don't know.
Or maybe click on the link. Yeah.
What? You changed the name.
Yeah. I mean, just to make it consistent.
It's still private.
No. So if you go down here. Oh, it's all ISMB 2000.
Yeah. It's my only. Can I change it back?
Okay. But it will ask for the. Okay.
No.
Is it public now?
Okay. It's downloadable from GitHub as well now.
So just a quick announcement. If you go on the GitHub and BHK Lab/ISMB 2019, you can download this material from GitHub also, which is a little bit more updated in terms of slides. Otherwise, it's more or less same info thing.
Thank you, everyone.